In our last lesson, we looked at the overview of going from a gene to a protein. In today's lesson, we're going to look specifically at the transcription stage. So that's where we go from DNA, the gene, to the messenger RNA. We're going to jump right in. The first stage of transcription is initiation. So this is where our RNA polymerase, so again, it's a different polymerase, our RNA polymerase is going to bind to the DNA. And it is going to bind upstream of where the gene is. So for example, if the gene would be starting here in this section, just cut off, then the RNA polymerase would bind before it. So it's called upstream. There are specific sequences of DNA that the RNA polymerase recognizes and will bind to, and that's called a promoter sequence. So this is the nucleotide sequence that lies just before a gene, or upstream is how I refer to it, and allows for the binding of RNA polymerase. This is often will contain a high TA sequence, and it's called a TATA box. So if somebody in the class refers to it as a TA box, we're going to guess that they didn't listen to this, and we're all just going to yell TATA at them, okay, until they learn. And this is in eukaryotes. We have a high A and T sequence. That's why it's called a TATA, T-A-T-A. And again, it's recognized by RNA polymerase. Why? Because there's less energy needed to break the 2 hydrogen bonds between A and T versus the 3 hydrogen bonds between the G and C nucleotides. So it's similar to the origin of replication. Prokaryotes have similar, we still refer to it as a TATA box, but it's T-A-T-A-A-T. So a transcription unit is a part of the DNA that is going to be transcribed to RNA. So we kind of loosely refer to this as the gene. The next stage is elongation. So this is kind of where the action is occurring. The RNA polymerase doesn't need a primer. So we don't need a primase to lay down an RNA primer. It can just start laying down complementary RNA right away. So the RNA is made in a 5' to 3' direction, just like the DNA polymerase 3 works. And this uses the 3' to 5' DNA strand as the template. So again, it's adding to the 3' end of the growing RNA sequence. The coding strand is the DNA strand that is not being transcribed. So this is just terminology. Why is that? Because it actually contains the same sequence as the new RNA molecule, except thymines would be uracils. The new RNA winds with the DNA template, and we get to see this in the next a little bit further in our slides, not in the next one, temporarily, but it will unwind as it extends, and the DNA double helix will reform. So we see here, let's look at this little part here. We have this big enzyme complex, our RNA polymerase. So it unwinds and it adds the complementary RNA sequence nucleotides to the template DNA. So this is the template that's being copied. This strand up here is going to have the identical nucleotide sequence as this RNA strand here, because this is complementary to this strand, and this RNA is also complementary to this strand. So that's why this is actually called the coding strand. So it's almost, I don't want to say it's a misnomer, but it's counterintuitive. We would think this is the coding strand because this is what's being read and leading to the RNA. But this is considered the coding strand because it will have essentially the same sequence as this RNA. So once RNA polymerase is well into the RNA elongation, another RNA polymerase might bind to the promoter and start a new RNA transcript. So if there's a high need for a specific protein, then you're going to want, the cell's going to want a lot of mRNA, and so it's going to have a quick production of that mRNA. The last stage is the termination stage. So you can imagine it's the end of that mRNA transcript. So RNA polymerase recognizes a termination sequence in the same way that it recognizes the TATA box. In prokaryotes, the proteins bind to mRNA, stopping transcription. Or the mRNA will actually fold on itself, creating a hairpin loop like we saw with what forms in the tRNA. In eukaryotes, one sequence is a string of adenines, which transcribes to a string of uracils. So think about that. On the DNA itself, there's going to be a bunch of A's, and it's called a poly-A tail. Poly meaning plural or plenty, and then A obviously is the adenines, and it's called the tail because it's at the end. So we have a poly-A tail, which is going to be read as a bunch of U's will be put in that place. And then proteins bind to the polyuracil site and stop transcription. A new RNA dissociates from the DNA template strand once it is completely done. So again, we have our image here. We have our RNA polymerase unwinding and laying down RNA nucleotides. And if we zoom in here, we have our RNA that is dissociating or unwinding from that DNA template. DNA being in purple, the green is the RNA. And we have our new nucleotides being added to the 3' end of that growing RNA. And remember, we have the growth is from 5 to 3' of the growing RNA strand. So some modifications have to happen. If you remember from our last lesson, it was considered a pre-mRNA until some modifications happened that I didn't get into. And then it became a messenger RNA, a functional mRNA. So a term to remember is until those modifications occur, it's considered a pre-mRNA. They are vulnerable to enzymes in conditions outside the nucleus. So remember, this is all occurring still inside the nucleus. Once it's been modified, it can leave the nucleus through the nuclear pores. Modification must be done before leaving the nucleus. Otherwise, there are enzymes that are more likely to degrade the mRNA, and you're not going to be getting the production of the proteins. So the first one mentioned is the poly-A tail. So not only does that function as a stop or a termination of transcription, but it is also helping with protecting that RNA once it leaves the nucleus. So it's pretty variable. We have a chain of 50 to 250 adenines, so it's not just like 5 as shown in the diagram. And this is being added to the 3' end because it's going to be the end of that pre-mRNA. So there's an enzyme called the poly-A polymerase, and this adds one adenine at a time. The tail prevents RNA digesting enzymes from attacking in the cytosol. And this is called polyadenylation. 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 Yes, I said it 3 times. Make sure you say it right. So why would there be RNA degrading enzymes? So think about that. Why would we need RNA degrading enzymes? We're going to talk about that in class tomorrow. And I want you to think about why we might actually want to degrade RNA in the cytosol. The second modification is the addition of a 5' cap. So the poly-A tail is put at the 3' end, so the end, if you will, so it's the last thing added to the mRNA. The 5' cap is going at the start of the mRNA, the 5' end. And it is specifically 7 guanines. So the poly-A tail is quite variable, whereas the 5' cap is specifically 7 guanines. This site acts as being recognized by the ribosomes, and the ribosomes are going to use it as the site of initial attachment to the mRNA. And we'll look more at this in our next lesson, but we had a little animation showing how the small subunit of the ribosome meets up with the mRNA, and now we kind of have a better idea of how that a small subunit recognizes where to bind or what to bind, and that's the 7 guanines that are at the start of the mRNA. The enzyme that adds the guanines to the pre-mRNA is called guanyl transferase. And just showing here, we have our pre-mRNA that is getting its 5' cap added, and that's again by guanyl transferase, and it has its poly A tail. Here we go, our 5' cap of 7 guanines to the mRNA, and we have the poly A tail. And we see these ... because, again, there's variability on how many adenines are in that poly A tail. Okay, lastly, we have introns and exons, and this is a really exciting area. I love this section of modifications of genes. So eukaryotes, unlike prokaryotes, contain coding and non-coding regions within a DNA sequence. So if we look here, here's our DNA sequence. And so this is what we would refer to as a gene, for example. What's cool is there's sections of this gene that are exons and introns, exons, introns. And notice they flip-flop, so exon, intron. This whole DNA section is going to be read and transcribed into pre-mRNA. So you're going to get your mRNA or pre-mRNA produced with both exon and intron, so exactly as it is. An exon is a sequence of DNA or RNA that codes for part of a gene. So the exons, despite its name, I feel like X should be exited. Exons are the important coding parts, and I say that with apprehensively, but they are what you in grade 12 might consider the important parts that are going to help code for proteins. The introns are non-coding sequences of DNA or RNA. So it's often referred to as junk DNA, which breaks my heart, and we're not going to do that. So for example, we have this exon here, which is coding for a protein, and this is the intron that isn't actually coding for the protein. This exon is coding for the next part of this protein, and this would get almost skipped over, and we're going to show how that happens. And then this is the rest of the gene, this would be removed, more of the gene, this would be removed, and last part of the gene. So prokaryotic DNA does not contain introns, so it's just eukaryotes that have this. And you'll learn more about how important introns are in protecting the genome. So introns are interspersed among the exons and are transcribed into mRNA, or pre-mRNA, I should say. So like I said, we have our DNA, and that's going to be transcribed into the pre-mRNA shown here. So it's exactly as it is in the DNA. Introns do not code for any part of the protein, so they ultimately do need to be removed. So this is why they're called junk DNA, because they're not actually coding for the proteins. So ultimately, this section of the RNA is going to get removed. And then these get brought together, and we see how it's missing in the final mRNA. Similarly, this intron would get removed, and we see it missing here. Same for these two introns, missing and missing. Okay, so those get removed for our final mRNA product. So how does this happen? So the word excision means to remove. So if we are excising DNA or RNA, we are removing that RNA from the product. So mRNA splicing is where introns are removed in the nucleus, this is still happening, from the pre-mRNA. A spliceosome is the enzyme protein complex, so it's enzymes and other proteins, that removes the introns. And then we have these cool things called SNRPs. SNRPs, yeah. These are small ribonucleoproteins, so think of RNA, nucleoproteins. So it's an RNA and protein complex. These bind in a particular order to the pre-mRNA. And we're going to look at how spliceosomes and SNRPs work together to remove introns from the pre-mRNA. So let's look at this diagram first. So we have our exons, 1, 2, and 3, and we have 2 introns that need to be removed. We have our 5' G cap, and we have our poly A tail. Once these introns get removed, and then we splice, well, it's called splicing, and then we ligate this DNA together, RNA together, we get our final mRNA product. So this is the picture in your textbook, and we're just going to walk through the diagram together. So first we have our pre-mRNA shown here. We have an exon with an intron and then another exon. We have our 5' end and our 3' end. So we would have our G's down here and our poly A tail here as well. So first we have our SNRPs recognize and form complementary base pairs with the mRNA. So here's our SNRPs in purple. And again, remember these are made of RNA and protein. So they are going to recognize and form complementary base pairs with the mRNA sequence at the junctions of the intron. So if we look at it straight here, they are going to be forming complementary base pairs here and here. Other SNRPs are recruited, causing intron to loop and bring 2 exon ends close together. So here's an exon end and here's an exon end. So now they're close together. You can almost imagine we're just going to have some cutting of that looped up part, and then they're just going to bring those 2 exons together. An active spliceosome, so remember that protein named spliceosome, is going to cut the intron and splice the 2 exons together. So it's a little bit different from having something like DNA polymerase 2 cutting and removing things and then ligase coming in. This spliceosome complex is going to do the cutting and the ligating all at once. Okay, and then we have the released intron, which is degraded. So back to my original question. And these SNRPs can go off and help excise another intron from a different pre-mRNA. So alternative splicing is a really cool thing that happens with pre-mRNA. So alternative splicing means that there's different exons can be joined in different combinations. And this is going to end up forming different mRNA. So you might not just remove an intron, but maybe an exon or a couple exons are going to be removed, and that's going to produce a different mRNA. And then you can imagine that's going to produce a different polypeptide. So 3/4 of all human genes are subject to alternative splicing. This is how we can get 100,000 proteins from only 20,000 genes. So the alternative splicing allows more variety of proteins to be produced. Typically, splicing produce a family of related proteins. So these proteins are going to do similar functions or be involved in a similar metabolic process or functional structural families of proteins will have a identical pre-mRNA, and then just certain different exons will be spliced out. So that is our nice quicker lesson where we went over the stages of transcription, where we have initiation, elongation, and termination. And then we have our 3 types of modification of the pre-mRNA before it can leave the nucleus. So we had the addition, we had the poly-A tail, we have the addition of the 5' cap, and then we have the removal of introns.